So our first speaker of today is Dr. Victoria Shabadina. Victoria has done her PhD in biochemistry in Münster, and before she shifted her research focus to evolutionary biology and the origin of animal multicellularity. Victoria now works at Barcelona at the University of Pompeu Fabra, where she studies the connection of multicellular organisms and the wonderful secrets of the unicellular holozoans, relatives of animals. So Victoria will tell us now more about these holozoan helpers and how animals appeared on Earth.
Thank you, Nina, for the introduction, and hi everybody. Thank you for joining my talk today. So I would like to tell you about what and how we study in our lab in Barcelona. We study evolution of animals, in particular the very beginning of animal history on Earth, the period when first animals appeared. You might know that hundreds of millions years ago, which is very, very long ago, the Earth was inhabited only by unicellular organisms. That means these living creatures were made only of one cell. But something happened 600 million years ago, and the first animal appeared. It was a small and simple animal consisting of probably dozens of cells, but in the millions years to come, it evolved in the variety of shapes and forms of animals which we can see now on Earth, including humans, because we are also animals, of course. So actually, how multicellular we are? One human body consists of 37 trillion cells. Not million, not billion, trillion. This is 4,000 times more than the number of people living on Earth now. So you can imagine the scale, it's really a lot of cells. And unfortunately, people not always live in peace on Earth. So think how hard 37 trillion cells should work to make one human body happy and healthy. Of course, they should follow some multicellularity rules. And two main multicellularity rules is collaboration and altruism. That means that each cell in our body knows about its neighbors and work in agreement with these neighbors. And altruism means that each cell in our body not only thinks about its own well-being, but about the well-being of the whole organism. And it can sacrifice itself for the good of the whole body. So we want to understand how cells evolved to follow these rules in a multicellular body. And for this, we want to go back 600 million years ago to this single-cellular organism which gave rise to the first animal. And we call this single-cellular organism unicellular ancestor of animals. So you might wonder why we want to study past of animal history, not present. It is because if we know how big and complicated animal body came to be in the perspective of evolution, we can better understand how it functions. For example, if we want to know how a computer functions, or we want to repair the computer if it's broken, we should know how the computer was built from the very first wire up to the assembly of hundreds of details. And similarly, that's why we want to learn how the animal evolved from the single-cellular ancestor. So how do we do it in our lab? We cannot use the methods which scientists who study dinosaurs use. Because first of all, this single-cellular ancestor lived much longer before than dinosaurs on Earth. And secondly, it was small, soft cell, and it left almost no traces. But fortunately, there is a way which nature gives us to travel back in the time. And the key to this is hidden in every living creature on Earth, and also in you. This key is our genes, our genetic information. Humans have 20,000 genes, and these 20,000 genes help our body to grow, to develop, and to be how it is. So what we do, we compare genes of animals with the genes of unicellular relatives of animals and try to reconstruct how this unicellular ancestor looked like. It is in a way similar to our family history. For example, let's say we have two grandchildren. One of them has black hair, another blonde hair. That means that color of the hair is individual feature of each grandchildren. But let's say both of these grandchildren have green eyes. That means that the color of eyes, green, likely was inherited by both grandchildren from their grandparents, grandmother or grandfather. So by comparing grandchildren, we can reconstruct how grandparents look like. Of course, we know how our grandparents look, but imagine if we didn't, this would be the way to reconstruct it. So we do similar thing. We compare animals, one grandchild, to their unicellular relatives, second grandchild. And by comparing, we want to reconstruct how unicellular ancestor looked like and how it functioned. And we have these unicellular relatives in our lab. We keep them in such flasks with nutritious liquid inside, and they can grow in our lab for years, so we can study them. Of course, these creatures are really small, and you can see them only under microscope. But to give you imagination how they look like, me and my colleagues in the lab, we prepared a few models of these cells. I will start from this one. So this is called salpingoeca. I think very nice, interesting name. So this is one cell, and salpingoeca has this long thing called flagella, and then flagella rotates in the water where salpingoeca lives. It creates a swirl of water, and bacteria also live in this water. Following the swirl, go inside the collar of salpingoeca, and that's how this organism feeds on the bacteria. This is the bacteria. So this is the first one. The second one, which we work with, is called a capsospora. Capsospora, it's also one cell, and it has long legs. They're called philopodia. And it uses these legs to walk on the surface similar to a spider. And the third model we did is called pyrum. So actually the cells I showed you, they don't have color, but we painted them to make them more attractive for you. But pyrum, then you look at it under the microscope, you shine a light on it. And pyrum cells reflect light and with the different colors, like a rainbow or diamond. So basically under microscope, it looks more or less like that, how we painted it. It's very colorful. It's a very beautiful thing to see. So back to genes. Then we compared the genes of animals, and of these guys, we came to a big surprise. It appeared that many genes which we thought were specific to animals, they're actually present in these organisms too. For example, if we again go back to salpingoeca, salpingoeca can hold on the other cells in the environment there it lives and form colonies. So the gene it uses to hold on each other and form colonies is the same gene which animal cells use inside of our body to hold on each other. And this is pretty amazing because this gene is only present in salpingoeca and in animals and in no other creature on Earth. That means that this gene was inherited by both animals and this organism from the unicellular ancestor, the guy we're exactly interested in. So that's how reconstruction of ancestor by comparison of gene works in principle. Another example with capsospora, this one. Capsospora has another gene which in animals helps embryo development, which is development of a baby inside of mama, which is also pretty surprising because embryo development is very specific to animal thing. We don't know now what this gene does in capsospora, but we work on it, and then we understand its function in this organism, we also will be better understanding the embryo development in animals. So what is the home take message from my story? First of all, I think it's pretty amazing how nature, how evolution work. Basically, animals didn't invent too many genes. They just took the genes from the unicellular ancestor and reused it and adapted to their own needs. And this is kind of make us not that special. Well, of course, animals and humans are special, but as much as these guys, because we all share the same miraculous mechanism of life. And that also means that we should respect every living creature on Earth, even those which we can only see under microscope, because after all, we are not that different. I hope you enjoyed my story and happy to answer any questions if there are.
Thank you so much, Victoria. Wow, that sounded really, really cool. So personally, I didn't even know how many cells we have in our body and how much we have in common with like animals and those funny little things you prepared for us. So I'm sure our audience has just as many questions for Victoria as I have. So this is another reminder to just drop your questions in the chat and get the chance to win a special prize awarded amongst all people asking questions. So I was actually wondering, like you showed us now 3 different types. Are you studying especially those 3 in the lab, or do you have a lot more?
We have more. We have around 10, 15 organisms from the same unicellular relatives branch. And so there are organisms which we study more extensively, and these are the 3 I showed, but we also have much more. We are also working on them. So actually there are more and more secrets which will be uncovered soon.
Okay, thanks. So another question we just received from the audience. To which non-mammalian aquatic organism are we most closely related?
So, aquatic. Yeah, this is actually the still not clear. So there are sponges which live on the earth. It's animals which are called sponges, and they are considered as the most primitive. I don't like word primitive because, as I said, every organism is special, but they are the most simple animals, and most likely, this would be the basic animal in our tree.
Okay, thank you so much. So another question. Will your study help us understand the variabilities or the individuality present in all the organisms?
I wouldn't say individuality of the organisms, but so another interesting thing about multicellular organisms, like also us, that we have different types of cells. Cells of the heart, of the eye, of the skin, and that's called cell differentiation. And our study will help understand how cell differentiation also evolved and led us to such a diversity of organs, of animals.
Okay, so thanks. Komal asked a question on Facebook. How do you find a common ancestor?
So ancestor, that's how I explain. So we compare genes of the unicellular relatives to animals, and by comparing genes, we see what is similar, what is different. The similar genes, which present both in the unicellular relatives and in animals, that means that they were inherited from the ancestor. And that's how we can, by knowing which genes the ancestor had, we can reconstruct, not 100% precisely, but quite precisely how it functioned.
So you basically assume that they have the same ancestor, and by comparing those two, you're trying to figure out what the ancestor looked like.
Well, we don't assume, we know, because again, as we found the genes in these guys, which are the same as in animals, and they're only present in these guys and in animals, that means that we come from the same ancestor. And that's how we decided that we want to reconstruct how this ancestor looked like.
Okay, thanks for the clarification. Natalie asked on YouTube, are there any unicells which exist in humans but not in any other animals?
What unicells? I don't know what
I guess she means like unicellular organisms. But that's just assumption. Please, Natalie, correct me if I'm wrong here.
So I will give 2 answers. As I said before, we have a lot of types of cells, different types, and some types of cells, they are specific to animals. I'm not sure if there is some types of cells that present only in humans, probably not. And of course, we are full of unicellular things, mostly bacteria, because bacteria live on our skin, in our stomach, even in our lungs, and they help us to live also. So it's symbiosis with bacterial cells.
Okay, then another question on YouTube from Mart. Do you know when your models evolved?
Yes, we know, because also by doing some analysis with genes, which is called phylogeny, you can estimate the time when these things appeared. But I don't remember now when was it.
Okay. And also, Salmon asked on YouTube, how far can we go forward with your methods? Can we explain the social impacts on cellular changes?
Social impacts, well, I guess social impacts have its influence on our behavior. So in the complex system, which we already have nerves and perception and reaction, so it's not on the cellular level. So I would say what we study, it's more basic. It's more about how everything came together in us.
Okay. Then, if you have to name it, what would be the gene that can be characterized as most important to allow multicellular organisms to exist?
No, I cannot. There is no one single gene. And moreover, what we are coming now to a conclusion, it's not only the variety of the genes we have which makes us multicellular, but it's regulation of these genes. Then one gene is activated, another deactivated, how they interact between each other. It's called genomic regulation. And this, as we think, actually played the most role in making us unicellular. So regulation of usage of the genes.
Okay, so do those genes define how we are on 100%?
Yes and no. So the genes are actually kind of write us, how the shape of our body, our organs, how we function. But we are not isolated organisms. We live and interact constantly with the environment, with weather, with climate, with other animals and plants, with bacteria again, with humans we meet, and all this together makes us how we are. So the genes do the basics, but they don't control our desires, our wishes, and our behavior completely. They give us direction where we would go, but 100% no.
Okay, thanks. So the last question for today. Where do you get these unicellular organisms from? I mean, the ones that you culture in the lab.
Mm-hmm. Yeah, so they were found in different places on Earth. Actually, there are also scientists who just go into the wild and investigate what is out there, and they look at under the microscope on the water drops or some inside of some animals, and in this way they discover new organisms. So this organism, for example, lives in the seawater, and Capsospora lives inside of the fresh water snail. So they come all from different places, but what unites them is the genes.
Okay, so thank you so much, Victoria, and thank you to all of our audience for your questions.